Hello everyone, I'm Dr. Eris, and I'll be leading our session today. We are going to dive into a critical topic in epidemiology, hantavirus. We'll explore its biology, how it spreads from animals to humans, and what we can do to stay safe. To start, hantavirus refers to a whole family of viruses. The key thing to remember is their natural reservoir, rodents. Humans typically become accidental hosts when they come into contact with the waste or nesting materials of infected mice or rats. Looking at this illustration, you can see the viral particle. Historically, these viruses gained major attention during the Korean War. Much later, in 1993, a localized outbreak in the Four Corners region of the US led to the discovery of a particularly deadly strain. Depending on where you are in the world, hantavirus manifests differently. In Asia and Europe, it often causes hemorrhagic fever with renal syndrome, or HFRS. In the Americas, we more commonly see hantavirus pulmonary syndrome, or HPS, which affects the lungs. By understanding the structure and history of these viruses, we can better appreciate the public health strategies used to control them. Next, let's take a closer look at the specific rodents that carry these viruses. Building on our introduction, let's identify exactly where these viruses live in the wild. We call these host animals natural reservoirs, and for hantaviruses, those reservoirs are primarily specific species of rodents. As you can see here, the primary culprits are deer mice, cotton rats, and rice rats. What's fascinating from an epidemiological perspective is that these are asymptomatic carriers. This means the rodents themselves do not get sick. They can carry and spread the virus throughout their entire lives without any visible symptoms. The virus leaves the rodent's body through a process called viral shedding. It is excreted in their urine, droppings, and saliva. Once these materials are in the environment, they pose a significant risk to any humans who might encounter them. This brings us to a fundamental concept in public health: zoonotic disease. A zoonosis is any infection that jumps from animals to humans. Understanding this animal-human interface is the first step in preventing outbreaks. Next, we'll look at exactly how that jump happens through different transmission pathways. Let's explore the transmission cycle of hantavirus, moving from the natural reservoir to human infection. Everything begins with infected rodents, such as deer mice, which carry the virus without showing symptoms. The virus enters the environment through shedding in rodents' saliva, urine, and droppings, where it can survive for several days. Aerosolization is the most common route. This happens when you breathe in microscopic dust particles contaminated with rodent waste. You can also be infected through direct contact, like touching a contaminated surface and then touching your eyes, nose, or mouth. Once the virus enters the human body, it targets the lungs or kidneys, potentially leading to severe respiratory or renal distress. Building on our understanding of how the virus spreads, let's look closer at its internal blueprint. To understand how it causes disease, we must first examine its viral structure and genomic composition. The virus is characterized by a negative sense single-stranded RNA genome. In virology, negative sense means the RNA cannot be translated into protein directly. It first acts as a template to create a complementary positive strand. This genome is unique because it is divided into 3 distinct segments. These segments are categorized by their size: small, medium, and large, or S, M, and L for short. Each segment has a very specific job to do for the virus to survive and replicate. Looking at this table, we can see the functional breakdown. The S segment encodes the nucleocapsid, which is the protective protein shell around the RNA. The M segment is responsible for the envelope glycoproteins, which the virus uses to latch onto and enter human cells. Finally, the L segment encodes the viral polymerase, the essential engine the virus uses to copy its own genetic material once inside a host. Now that we've seen what the virus is made of, let's see how it puts these tools to use during an infection. Welcome. In this simulation, We'll examine the critical steps a virus takes to hijack a human cell, starting with attachment and ending with replication. Binding affinity measures the strength of the attraction between viral surface proteins and host cell receptors. Higher affinity makes attachment more likely. Receptor density refers to the number of integrin receptors available on the cell surface. More receptors provide more opportunities for the virus to dock. When both affinity and density are high, the virus attaches quickly, triggering endocytosis, where the cell membrane engulfs the virus to bring it inside. After entry, the virus undergoes uncoating to release its genetic material into the cytoplasm, where it begins replicating to create new viral particles. Notice how reducing these variables makes it significantly harder for the virus to successfully bind, illustrating how some cells are less susceptible to infection. Now that we understand the infection mechanism we just covered, let's examine the actual clinical impact on patients. Hantavirus presents as two distinct syndromes depending on the specific viral strain and geography. To give you a clearer picture of how these diseases manifest in the body, let's watch this short clinical comparison.
Pathogens like HPS can damage lung capillaries, leading impacts kidney function by injuring Sogwart Champisa.
As the video illustrated, Hantavirus pulmonary syndrome, or HPS, is the variant we primarily see here in the Americas. It specifically targets the lung capillaries, leading to rapid respiratory failure, which is why early detection is so critical. In contrast, this table highlights hemorrhagic fever with renal syndrome, or HFRS, which is more common across Europe and Asia. While HPS attacks the lungs, HFRS focuses on the kidneys. You can see the variations in severity and symptoms here, which are essential for clinicians to distinguish when a patient presents with a suspected infection. Now that we've examined the biological impact and the differences between HPS and HFRS, let's translate that knowledge into action. Based on the public health response we just discussed, we need to focus on how to actually prevent infection. This page outlines our most effective tools for staying safe. The first and most fundamental step is rodent control. Since rodents are the natural reservoir we identified back on page 2, keeping them out of human environments is our primary line of defense. You must seal even the smallest entry holes in buildings and secure all food sources. If there's no food and no entry, the risk of transmission drops significantly. Next is safe cleaning. This is where many people accidentally expose themselves. Remember our lesson on transmission pathways? The virus is most dangerous when aerosolized. Never sweep or vacuum dry droppings, as this kicks the virus into the air. You should wet down the area with a 10% bleach solution first. Let it soak to deactivate the virus before wiping it up. This simple step prevents the virus from entering your lungs. Finally, when dealing with heavily infested areas, you must use personal protective equipment. An N95 respirator is essential to filter out any particles that might become airborne despite your best efforts. Before we move to our final summary for the day, let's think about how we can share this information with our communities effectively. To wrap up our class today, let's synthesize the core concepts we've navigated regarding Hantavirus. It's important to see how these pieces fit together for community safety. First, the pathogen itself. As we analyzed earlier, Hantavirus is a rodent-borne virus. Depending on the strain, it can lead to severe respiratory distress or renal failure, making early identification of the reservoir key. Regarding transmission, remember that it occurs primarily through the inhalation of aerosolized rodent waste. This is why we must be extremely cautious when cleaning or entering spaces that have been dormant. This leads us to our most critical takeaway: prevention. As we just covered, effective environmental control and rodent management are far more efficient than any medical intervention after the fact. Ultimately, proactive hygiene and staying informed remain your best defense. You've all done an excellent job today. Thank you for your focus and your commitment to public health.